All right, let's finish up our little stories about hemoglobin with one last hemoglobin story, probably the most important. This is actually a big part in founding of the field of biochemistry, figuring this story out, because it has to do with a debilitating disease that is genetically associated and how we can treat that disease. Why is that disease around? So here's the first images of it. I've already mentioned what it is, so you probably already know what it is, but if you don't know, here's the first images that people saw, that this disease was a problem. In 1910, a Chicago doctor was investigating the blood of a dental student at the local university. The dental student was an international student from Grenada and presenting with anemia. So some sort of anemia. Looked at the blood, and you see something, even under the power of microscopes in those days. Very clearly there's something wrong here. There's a lot of sickle-shaped cells, misformed cells, in the blood. It turns out that they pretty quickly found out these cells are more fragile, and that's causing the anemia. The cells are breaking apart because there's something structurally wrong with them. So the thing about sickle-cell anemia, I have to mention a little bit of genetics right here, is that you have 2 copies of every gene, one from your father and one from your mother. So that means depending on what they carry, they each carry a hemoglobin allele of the particular genes for hemoglobin. You'll get 1 copy from each of them. If they have the same gene, then you will be homozygous for that gene. You'll make all that gene. But usually you have 2 at least slightly different genes, and you'll make 2 different copies. You'll be heterozygous. Genetic diseases, this saves us from a lot of genetic diseases, because a lot of times there will be a problem that will occur when you have all of one type of protein that's broken in the same way. That's what happens with sickle cell anemia. Somebody heterozygous will actually be protected from malaria for a particular allele of hemoglobin. But somebody homozygous for the same allele, the sickle cell allele, will have sickle-shaped red blood cells as a minority of their cells, but enough of a problem to cause anemia and associated medical symptoms. The thing about the protection from malaria, it was figured out from one clue. For example, there's a very geography-associated incidence of this gene. There's so much of it in Africa that 1 in 12 Africans are heterozygous, and even I can do the genetics math for that. 1 in 144 children of these Africans will have sickle cell anemia as a result. The sickle cells are definitely misshapen. They're not always sickle-shaped. Notice that the one on the right has some weird sort of spikes to it. So that shows you that something's going on inside the cell, and it turns out that it's with the proteins in the cell. Red blood cells don't have that many things to go wrong, and it turns out this is a problem with the most prevalent protein inside the red blood cell, a problem with hemoglobin. So there's been a lot of work put into sequencing hemoglobin genes. We can now detect whether you have a particular mutation that will cause sickle cell anemia. We found 300 variants or mutations of the hemoglobin gene. These are called alleles when it's a major population variant. And just remember that you have the homozygous versus heterozygous issue. Sickle cell anemia happens in a homozygous situation for a particular mutation, and it zeroes down to 1 amino acid. So for the future doctors in the group, I would like you to know what the symptoms would be like, because that's what you're working from. It's normal anemia-related symptoms, the kind of things that you associate with an anemia. There's physical exertion is going to make you weak. You're going to be dizzy, you're going to have short breath. Your hemoglobin content of the blood will be low. Your iron content, therefore, will also be low. The sickle-shaped cells also do something extra in this case. They block capillaries. You look at those things and you can see how some capillaries, the red blood cell just has to squeeze through. These can't squeeze through them, so they either break or they stick in there, and that can actually cause pain and loss of organ function. It's important to know that these are very general symptoms, and so there's been a lot of times where sickle cell patients will go in for treatment and they won't be believed. because you don't have an easy test that you can do to find out. And the symptoms do tend to be, there are capillaries all over the place, and the symptoms tend to be rather general rather than associated with a particular organ or a particular part of a body. And I just want to point out that there is a high amount of pain with this. There's a unbelievable pain. One patient says it's like a jackhammer on your back, throughout your whole body, pain in your joints. Basically, there's a flare-up of it, and it has to do with things like osmotic pressure and the concentration of the red blood cells. But there are episodes where the anemia happens more, and once you get something caught up in your capillaries, they keep getting caught up, and it gets worse and worse, and the pain is all over. SCA patients often get called drug seekers because they're asking for pain meds. But my important thing is to realize you should believe them. They need the meds in this case. There's an 8-year-old that talks about how they often get fevers. That's one of the responses to the body to this sickle cell sort of attack or crisis. My head starts getting warm. My eyes sort of turn yellow. Feels like someone's squeezing my legs and arms. Very, very painful. So what's causing this pain is a genetic mutation in hemoglobin. The good news is, because it's such a focused genetic mutation, there's a possibility that with new technologies we'll be able to fix it. So I want you to listen to one extra video beyond this. I want you to go to this video on YouTube, which is Francis Collins in his 2017 American Scientific Affiliation lecture. I have a link to it on Canvas. And I want you just to watch, it's an hour long, I just want you to watch from 52:30 to 57 minutes. So just 5 more minutes. The whole talk is really good, but that's the 5 minutes where he talks about what is the NIH doing right now to combat this and to cure sickle cell disease. Once you listen to that, you'll be able to put this in context. But I want to tell you a little more about the nature of the disease. It's a very small mutation that at least is crucial for the disease. So hemoglobin S is the sickle cell allele. It's different from hemoglobin A, always in one place. If you look at it, glutamate has been mutated to a valine at position 6 of the beta chain. Well, lo and behold, my little friend here is the beta chain, and like I showed before, here's the N-terminus, it's blue, I don't know if you can see that, but right here is the glutamate that we were talking about. You can see it's glutamate because I can see the little oxygens. And so I'll bring this to class, you can pick it up and confirm if you want, but this is the mutation that causes sickle cell disease. It is nowhere near the binding site. The binding site's all the way down here. And so if you are talking about a mutation here causing a problem, it's not causing a direct problem with oxygen binding. It's more likely a problem with protein structure, because that's the part that you're going to be dealing with on the outside of the protein. Also, it's not at the interface, and so it's going to be exposed. Hemoglobin S differs from hemoglobin A in tertiary structure. So we just talked about primary structure. In tertiary structure, you actually have, when you have a valine here, it actually makes a little divot in the surface, and it makes a hydrophobic patch. There's a lot of other hydrophobic residues around it. And so it makes this whole thing have a sticky patch right here on the molecule. But then that sticky patch, the sticky hydrophobic surface, is repeated, right? Because you have 2 beta chains. You've got one up here, one down here. And that is why this is bad news. Because you have 2 chains of beta, you have 2 sticky patches on either end of the molecule. And if you think about what we learned about fibrils, that's how you get a fibril, an open-ended structure where you can have binding at either end, especially with non-specific binding like the hydrophobic effect will do. So what happens if you have a lot of hemoglobin S around? Then the strands, it'll actually form long strands. The strands will align and form crystalline fibers. And that will cause the sickle shape or the just plain weird shape of the cell. This is a figure you've seen before if my head's blocking it, so sorry about that. In quaternary structure, this is a quaternary change. And so we have hemoglobin S being different at primary, tertiary, and quaternary levels. This is why the sickle cells are such a problem. This is a picture of some capillaries. These are very, very small. Capillaries in muscle cells right here. Sometimes they're only one red blood cell wide, and the cell needs to be flexible to fit through these. Sickle cells cannot fit. One of the other issues that sickle cell patients have is that they end up, I got this from the NIH, they end up with kidney problems. Because the kidney is another place where you're trying to filter things out of the body, and you also have very small capillaries. So the kidneys get basically gummed up by all the sickle cells. They end up with small tubules that are blocked, and they become necrotic, which is basically they start to die. So kidney problems will result from people, all from this one little amino acid being the wrong amino acid. It's not even that blatant of a change if you look at the structure of the amino acid, but it's enough to cause the fibrils to crystallize. Because the sickle cells are so physically different from normal red blood cells, there's actually some hope for assays that will actually build on this difference. In fact, here's a simple assay that will tell how much of the sickle cell you have in blood, a blood sample. If you just put them in a glass tube, and this glass tube is filled with some kind of dense sugar solution, doesn't really matter as long as it's dense. And all you have to do is spin it down. And remember that centrifuges can be built out of like egg beaters or toys or 3D printers or things like that. You can have them hand cranked. This could work in the farthest parts of Africa if you just have a centrifuge and this little tube. You take the blood, you put it in. Before centrifugation, it's like that. After centrifugation, it drops down. I'll try to make sure that you can see this. Right next to me, or right next to me there's actually a line at the very bottom. After centrifugation, you get a lot of red blood cells at the bottom, and you get different levels of red blood cells. Basically, the whole blood gets pulled down, and it gets pulled down to the bottom of the tube. The denser the cell, the more to the bottom of it's going to be. And this is where I'm definitely going to have to show you some kind of reprint of this. But what it shows you is, at the very bottom of the tube, the red blood cells that are sickle cell are dense because they've got all these fibers in them, they're misshapen, they get pulled down to the bottom of the tube. If you have a lot of red blood cells in that sample that are sickle-shaped, they will show up as a line at the very bottom of the tube. I hope you can see it, but you can trust me that you can see little red lines at the bottom of the tubes that have sickle cell anemia. So remember that all of this physiological result is from a single point mutation, and it causes the whole sickle cell anemia problem. So there's the question of why does this bad thing happen, right? You know, why do bad things happen to good people? And in this case, there's actually a reason that we've found for it, because as a population, the sickle cell mutation helps the population survive. As bad as sickle cell anemia is, malaria is worse. And one sickle cell allele will actually protect you from malaria. Their first clue was comparing the maps of the prevalence of the sickle cell allele, HBS, and the prevalence of malaria. Look at those two maps, and they pretty much overlap. Like we said right here, 8% of the African population is a carrier for this allele, and it's especially concentrated, it's 10 to 20% concentrated, in areas where malaria is most prevalent. So that caused theories. Malaria is a red blood cell related disease. Could it be that a little bit of the hemoglobin S in your system could actually prevent malaria from spreading? That used to be a question. Now it is a statement, because in my view it's pretty much been proven. I'm going to show you a couple of papers that say that. This is the one, well, it says it right here. Sickle hemoglobin confers tolerance to Plasmodium or malaria infection. So you can look up these if you want more information. The main thing is after they showed that it really was the fibers and that really was protective, they had this paper that came later that says, how does it actually work? And they show that there's actin remodeling that the malaria parasite forces in the red blood cells. It forces them to adopt different shapes, and they remodel the actin cytoskeleton as a result. Hemoglobins S and C, actually, is another sickle cell related allele, I believe. they actually will interfere with this process and make it harder for the malaria to reproduce inside the red blood cell. I think they have just enough of the fibrils forming that it gets in the way of the malaria, but not enough to cause the full-blown anemia. There's actually trafficking networks that move the Plasmodium proteins around. These sickle cell fibers interfere with those trafficking networks. That's all cell biology. You'll have to ask someone else about that, but I can tell you the sickle cell, I can see how sickle cell fibers could mess that up. And in fact, one of the things about selection for this has happened before is that selection for this is still happening. This trait is actually being selected for in a Darwinian fashion for this one particular gene. Realize that this is a gene where the population is caught in a trade-off. And the trade-off actually ends up good for the population because malaria is such a problem. Now that we're dealing with malaria with pharmaceuticals in another way, we can also use our pharmaceuticals to help sickle cell patients. And so we can hopefully get around this system and actually improve the hemoglobin genes of the populations. We can't go very far and we can't always make things better, right? But in this case, we have a very biochemically defined thing where we can actually make the two diseases better if we have pharmaceuticals for malaria and things to treat the fibril formation steps. These are the kind of clinics that will need to be able to use both malaria therapy and sickle cell therapy. This is the kind of hospital that you have in Africa. I'm actually working with my Burundi friends right now to teach the biochemistry again. I'm teaching them biochemistry right now, and there's such a gulf between our contexts. It makes the task of teaching a lot harder for them because there's something that I'll say and I won't even realize that I'm not communicating. So it's important to remember the difference in context, but hopefully with effort we can bridge this difference. We can come up with therapies in this case, and teaching in my case, that will help bridge the gap to all the people in Africa who are suffering from these diseases and the doctors who will treat them. So that's what I'm just saying, a little bit of why I get up at 6 a.m. to talk to Burundi, and I'm going to do that tomorrow. So don't come calling at my house at 6 a.m., but at 7 a.m. I think I'll be able to answer all your questions, okay? So how do cures work for sickle cell anemia? It's interesting that this molecule right here was one of the earliest sickle cell cures. If you look at it, it's actually familiar to a molecule that we've already seen. It looks a bit like urea, but it's hydroxylated urea. And what they found out, and I think it seems like one of those things you just find out by accident, hydroxyurea will actually stimulate the production of fetal hemoglobin, the HbF gene. We've all got it because we all were fetuses once and we needed to have that fetal hemoglobin with the extra affinity for oxygen to be able to breathe. So think about why HbF would help. Number one, you always have it. And number two, it would help because of what we already said about what it does. If you just have more of that, if you just turn that gene on, then you will actually help the anemia. There's two short readings that I've got. So there's the first thing, and these are also about cures. This is about using CRISPR to possibly cure sickle cell. It goes with the Collins lecture. And then we have a reading about glutamine as a new drug. This is a news summary sort of article. It's not a journal article, but I want you to read this article. It's complicated, and you'll see that there's a complicated story here. Good. Because our journal article for this quarter of the class, is going to be looking at the actual paper and the actual patent for why can you use glutamine to treat sickle cell. Okay. This is a figure about the CRISPR. If you know about CRISPR, it seems like a prime candidate to go after sickle cell. We've got one nucleotide that we want to correct, and if we can correct that in the bone marrow, then all the rest of the blood cells that will be made will have the glutamate in that position rather than the valine. And that is what I wanted to tell you about sickle cell. So realize that we sort of finished the chapter about hemoglobin right now. This brings it all the way out to a direct medical application. Don't forget to watch the Francis Collins lecture section and to read the two short readings. it would be helpful for you to do that before we get too far into Chapter 6, because Chapter 6, we're going to be talking about how enzymes work, and we're going to be getting into the data from the second of these readings, which is the glutamine reading. And with that in mind, on Friday we'll have a reading quiz because we'll be jumping into Chapter 6. All right. I will see you then.